Right. Let's move on to the little bit the 4 histone protein what I said. H2A, H2B, H3 and histone 4. And the other fifth protein is H1. That's a linker protein, which is essential for the chromatin condensations. Out of these all the 4 proteins, there are variants. Variants has specific functions. So the histone 3, it is a general. Most of the chromosomes will have the histone 3 proteins. Similarly, most of the chromosomes will have the histone 4. Histone 4 does not have a variants or isoforms. And H2A, this is a major form, and there are other variants are present in our cells. And H2B, no variant. So basically, histone 4 and histone 2B does not have a isoforms, no isoforms. Whereas H3 and H2A do have isoforms. It has isoforms, and that has very specific functions, very selective function in the eukaryotic cells. Coming to the linker protein, which has 7 isoforms. I am not going to explain in detail. Mostly all the isoform has a same functions, very slight differences, but largely it helps to bind with the 2 nucleosomes, between the 2 nucleosomes of the linker DNA here, and helps to fold or compact the 2 nucleosomes together, primarily for a condensation. That is the function of H1. So the histone 3, it has a 2 important variant. One is 3.3, which is almost similar like the original histone proteins. Only few amino mutation here and there, which is not having a very kind of very selective functions. Even the function of these variant is not very clear. The histone 3.3. The another histone 3 variants, that is a SNPA, which play very important role, and mostly this is present in the centromere chromosomes, this NPA. And if you look at it, the common, the structural organization of all the histone protein, there is a domain called HFD, right? Histone fold domain. So this is the domain responsible for my histone core, obviously N-terminal regions. So the HFD is a considered across. So the SNPA is a key variant present on the centrosome, the centromere. See, coming to this histone 2A variant, the 2 important variant, H2A.Z and X. I'll explain this about a little bit, and also the macro H2A. Okay, so if you look at it, the H2A and then A.Z, there is a 2 amino acid lesser. You can see the number of amino acids here in this case, 135, 135 in this, and then here 133 lesser. But in H2A.X, very important histone variant. I'll explain why it is so important. It has some few additional amino acids. Another important variant, that's macro H2A. It's very unique. You can see it has a excess amino acid in the C-terminal regions. It is also it does not present throughout chromosomes, but selectively on a few chromosomes, it has a very important function. I'll tell you. So these are the different histone isoforms. Particularly, H4, H2B does not have isoforms, and H3, H2A do have isoforms. Right. So the histone 3, then the first isoform I said 3.3, and it is actually replaced by one of the histone chaperone called histone regulator, HIRA. As of now, the study shows, although sequentially very similar, but this presence of this histone 3.3 mostly on the actively transcribing genes. So most of the active chromatin will have this histone 3.3. And along with the another important variant called H2AZ. So I said 2 variant of H2A, A.Z, A.X. A.Z is a euchromatin histone. That means actively transcribing genes will have, instead of H2A, this will be replaced with the H2A point Z. So basically this is the euchromatic histone. Why it has to do, why cell has to do? No, you can think about it. So if instead of just changing the modifications, so many modifications need to change. So for that purpose, sometimes the specific region cell will simply replace the whole histone to the another new variant. That will take care of the expressions. So instead of recruiting the demethylases or methylases, removing, so this is a lot of process. So for that purpose only, the variants are evolved, can quickly replace with the whole histone which has any. For example, the H2A will have some kind of repressive modifications which need to be erased or then new modification added, so that is double the process. So minimize these. So it's possible the cell will simply remove the whole H2A, repress H2A, and then bring the active. So this is what. So but for that again, it doesn't do just like that. It needs a lot of factors. That's chromatin remodeling factors and then chromatin assembly factors, which I'll briefly tell you. The macro H2A, it is an important variance, mostly present only on an inactive chromosomes. So basically, macro H2A is a repressive histone variant. So H2A point Z is a active histone variant, and macro H2A is a repressive histone variant. Mostly it is present on the X chromosome inactivations. So it, as of now, the data shows that the macro H2A is highly rich in the highly condensed X chromosomes, in a female X chromosomes. So that also tells that the macro H2A participate in the X chromosome inactivations. The SEN-PA is another histone variant. As I said, it's specific to the centromere, and more importantly, it is essential for the kinetochore and chromatid separation, which makes sense. So yeah, so this is how, this is the place you have the histone variant, the H3 variant, what I mean, okay, H3. So I already explained histone 3.3 for a euchromatin, and in this case SEN PA variant for the centromere regions where it is present, and you can see here the DNA stain, and then this is a SEN A staining. You see mostly present only in the centromeric regions. You can see this color, right? And this is the regions, this particular histone variants highly present, and all other regions you have the normal H3 or H3.3, right? So only this region will have this particular histone variants. You see, this mostly present nearly 0.1 to 1.5 megabases. So the 1 megabases, I hope you know that. So you'll have one base, right? One base pair, and then next is 1000 base pair, that we call 1 KB, right? Then next immediate nomenclature after 1 KB, this is 1000 base pairs. So 1 base pairs and then 1 KB, that's is equal to 1000 base pair, right? The next is 1 MB, that's 1 megabases or 1 megabase pairs, which is 1 million base pairs, 10 lakhs, right? And then next is 1 GB. So these are four major terms you should keep in mind. So 1 giga base pairs, this is 1 billion 100 crores base pairs, right? So the average length of these centromeric regions roughly around 1.5 million base pairs, where these particular variants are occupying. That's information. right. So I explained about histone 3.3 very briefly, and then cent A variant, which is for centromeric regions, and also explain H2A point Z, right, and that also explain H2 as a macro H2A. So these are the variant I explained. Another important variant that is H2A point X. It's a very important variant, very unique. So this is histone variants. It's get immediately replaced wherever the cell will have, or it gets a double stranded DNA break, DSB. So basically this is a histone variant is a marker for the DNA double stranded break. So nearly up to 2 to 25% of the histone pool this actually present, because that also says that cell undergoes a lot of double stranded breaks, and then it is a marker, and then it helps for the repairing of the double stranded break. When cell undergoes for double stranded break, this is a very important variant comes and takes care of it. It is site for the double stranded break. Later the double stranded break machinery comes and repairs it. And there is a one particular residue at 139 serine which gets phosphorylated in the H2A. So the phosphorylation of the H2A point X at this position is very important site or marker for the DSB, double stranded break in the cells. And this phosphorylation is done by P13K like family of kinases. And more importantly, what happens, there is a protein called MDC, that is a mediator DNA damage checkpoint proteins. So this is a protein which actually monitor are there any double stranded break in the cells. It's a very important protein for DNA repair machinery, and then which helps for this recruitment of the repairing machineries. So MDC protein is it's a protein which monitors the double stranded break. So what does it do? It binds to this particular variant via it is the C-terminal domain. So now you see how this is connected with the double stranded break. So what happens when you have a nucleosomes, right? And then there is any double stranded break here, and cell immediately comes and replaces with the H2A point X, and then which gets phosphorylated. It's not very clear after integration it gets phosphorylated or phosphorylated form it will come and integrate. Phosphorylation of this particular serine, that information not very clear. So this phosphorylated H2A will get integrate. Then you have a 2H2A point X in this particular nucleosomes near to the double stranded break DSB. Then after that, the MDC protein which actually monitor or checks for the presence of double stranded break. How does it check? Particularly, it recognizes the H2A X, which is a marker for a double stranded break. Then it via it is a C-terminal domain. Then it recruits all the repair machinery, DNA repair machinery, and then repairs it. So this is how this variant functions. It's not exact epigenetic function. You see, it play very important role in the DNA repair machinery. Right. So now it's clear, right? So you have the I said H3 point. Let's go through once again the major variant functions. So H3, you have H3.3. This is for a eukaryotic regions where these get replaced, or active regions. Then you have CEN PA. This is for the centromeric regions. Is also clear. Now H2A, you have A point X and then A point Z, and A point X is for your DNA repair, and then Z for your again euchromatin or active regions, active genes. Then I said macro H2A. This is for inactive chromosomes, particularly X chromosomes. So this is the major function of the histone variants. Right. So you should know how this assembles in the on the free DNA molecules. So it's very important to know. When you understand this, then you will understand how the histone inheritance takes place post DNA replications. The lot of theory, again, it's not very clear evidences. This is all the informations based on the biochemical evidences in the in vitro assays, but in real, in systems and cell, how this assembly takes place is still active research actually. So what people have identified, it is a possible, we are in the DNA replication regions. Okay, you have the 4 histone protein H2A, H2B, H3, H4, right? Then the new histone need to be synthesized and then it has to be brought in for this daughter strand. So people have hypothesized these histones can break apart, all the 4 histone proteins can break apart and reassemble. That is possible. Or it is possible that the H3 as a dimer exists and the newly synthesized H3 will come. This will go for this one of the strand and this will go for another strand. So both the theory, at least based on the biochemical evidences people are proposing, and which comes first, so those are all the questions, again, is not very clear. But yes, so what is known as of now, it's disassemble in the replication foci and then it assembles to form a new nucleosome on this at the post replicative stage. Right, this is another theory they said. So as I said, this histone 3, the histone 3 is a homodimer. Histone 3, histone 4 are heterotetramer structures. These also may break down into species, and then based on these incoming new histones, because the mark need to be distributed. See, the epigenetic mark, whatever present, for example, repressive mark present here, that need to be added in the both, right, both the nucleosomes. That's very important. Then only you are maintaining the heterochromatic regions in your daughter cells, because more than 80% or 70% of the chromosomes are maintained repressed. That means these need to be repressed post replicative. So that means the mark need to be inherited. Similarly, housekeeping genes marks need to be inherited. So all the inheritance takes place by the combinations of assembly and then reassembly. So again, this the theory is it's possible, and the theory proposes it is possible all the histone can dissociate and then assemble it, where the combination can go in both the strands, H2A, H2B, H3 and H4, and then it's possible that it can stay intact and communicate a message to the incoming histone 3 and histone 4 complexes, and then the repressive modification also should be transferred to the new nucleosome and then should maintained. So that's how all the theories or biochemical evidences are proposing. Right. One should keep in mind. So I said the histone proteins highly basics, right? So it tend to interact with the DNA just like that, because of high affinity. DNA has a negatively charged, this is a positive charge, it can quickly integrate. So the unwanted integration is really bad for a cell, because it may hamper the DNA replications or normal growth of cells. So cell evolved with, there are factors, this is chromatin assembly factors. It is also called histone chaperones, which play very important role for a successful nucleosome formation post replicative. That's very important. Otherwise, the histone proteins can simply, because in the post disassembly, I said two theory, disassembly of these all the protein and reassemble. It's possible this protein can simply go and bind to the this replicated regions. If it binds, it may prevent even the replication process, right, which is unwanted to the cells. The cell evolved with these assembly factors. So basically, what does it do? It prevents the the histone interactions with histone isoform alone interaction with the DNA, which is unwanted. So what then cell needed is the intact nucleosomes. All the 8 nucleosomes need to assemble. So that's a place these chromatin remodeling factor play very important role. CAF is a chromatin assembly factor. CAF, you can see the number of subunits are 4. It primarily takes care of binding with the H3 and H4. That means it takes care of the individual H3, H4 does not go and bind to the DNA structures. That's very important, because histone alone can bind to DNA structures. To prevent these, this CAF will take care of it. Similarly, the RCAF, another important, that is replication this is a chromatin assembly factor, and then RCAF, that is replication coupled assembly factor. So these also play very important role to minimize the individual isoform interaction with the DNA structures. Definitely, if it interacts, when you give a DNA free, when you just take a DNA free DNA histone 3 alone or histone 4 alone, when you conduct interaction assays, you will have the very strong interactions. Right? This can happen in the cell as well. So cell evolved with the systems that okay, this kind of unwanted interaction should be minimized, which may hamper the DNA replication process and assembly and so on. So further, this factor comes in and takes care of it. So these also takes care of H3 H4 interactions. That means it sequesters these things and then does not allow as an individual component go and interact, and later these assist for the nucleosome assembly as well. And then NAB, nucleosome assembly proteins, NAB1. So this is an important protein for H2 H2B. So this is how the these are the key proteins at the replication fork. But there are histone chaperones called rungs, okay, and then Hira, I said, right. So these are the histone chaperones which works at chromatin level. So these works at the nucleosome, DNA replication fork regions, these chaperone proteins. But these histone chaperone proteins, what does it do? It can directly work at a single post-replicative at a specific genes. For example, you have multiple nucleosomes in a particular regions, after assembly, and a few of the nucleosome need to be activated or repressed. So the histone protein need to be removed and replaced. So that's the time this chaperone works out. So you see the histone chaperone function at different levels. All it helps in the proper assembly and replacement. So these should not confuse with these 3 important factors in the replication fork. Right. So this is the histone assembly, and then the histone 1 protein, important linker protein. You see, this is the linker regions, right? The linker regions are linker DNA, which is roughly 40 to 50 bases. So together one is nucleosomes with the histones roughly 200 base pairs. So the histone protein binds here and then helps to fold together. So that's why the folding is shown like this. After folding, we can imagine you have these, and then the histone 1 will be like this, folded with the DNA structure. Then only then another nucleosome can come closer, right? So this condensation is achieved through these mechanisms. Very important protein, essential to convert 30 nanometer fiber structures, right? I said the 7-fold 10 nanometer, that is most likely euchromatin regions. That time it do not cell do not have a real condensations, but to form a 30 nanometer fiber structures, that is the condensations, packing of nucleosomes to the typical compactions, this protein play very important. Without the protein, the nucleosome will not get condensed. That's very important. There are organisms does not have these proteins. There are some eukaryotes. So where the higher order euchromatin, heterochromatin structures, condensed chromatin structure, in fact, it's not there. Right. Just to refresh, the acetylations in general all play very important role in the gene activation by opening up the chromatin, basically reducing the charge on the histone proteins or neutralizing the charge on histone protein, because acetyl group carries the negatively charged, whereas the methyl group does not carry any charge, even the methylation additions to the histone protein does not alter the charge. The acetyl group alter the charge on the histone proteins. So just to show the packing unpacking is one example. Acetylation opens the chromatin and methylations closes the chromatin and the best example is repressive modifications. Histone 3, lysine 9, methyl 3. We will see this modification much more detail in upcoming classes. So, this is the repressive modification. That means chromatin need to condense, need not to access to the, or need not to give a space for the transcription factor. So, it assists for the chromatin condensations. How? We will discuss in this in a canine modification class. Right. So, this is the 30 nanometer fiber structure I said. You see, it is packed like this, and H1 contributes very important. I mean, H1 is a very important factor, contributes for the condensations. This is a one. There is one more which I will explain during acetylation classes, that also contributes for the condensation. But this is the major one. The first level of compaction is very important. So, you can consider this is an heterochromatic structures. See, the without H1 is a euchromatic structures. So, a lot of theory how this compaction happens. A lot of proposal came on because in a cell there was no clear tool or assays to show how this arranged. Because at the end of the day, it has to arrange, right? I said if you recollect the introduction, 2 meter of DNA structure need to be packed. That means it had to be packed very carefully as a chromatin or chromosomes. How this packing happened is not at all very clear actually. But there are biochemistry evidences, data shows that are proposed 2 models. The one is solenoidal model. So, you can see it is like just normal folding. This is a typical solenoidal model. You can see from the top view how does it looks like. But the other model, which is a more acceptable model, is zigzag model. Okay. So, the zigzag model of packing, this is a packing of nucleosome into chromatin. Basically, this is a structure you can imagine a 30 nanometer fiber structures. There are 2 model has been proposed based on the biochemical data. One is the solenoidal model and then zigzag model. So, zigzag model, how does it look like? You can see these 2 nucleosome structures. This solenoidal is says straight away packing. Here it comes like this and then it packed. So, the biochemical evidences confirms this is the likely compactions in a cell as well. Okay. Zigzag model is the most acceptable model that nucleosomes are packed in these cells. So, this is because that gives a more space compared to the one solenoid. You can see here there is a 2 nucleosomes, nucleosome 1 and then nucleosome 2. Similarly, here nucleosome 1 and nucleosome 2. So, it can in a zigzag model, it can more of these histone, I mean nucleosome structure can be beautifully packed. And yes, because this was accepted because the EM electron microscopic structures, the model proposes similar to the zigzag model. Of course, still people always doubt about it because this is the in vitro data. When the increasing salt concentration, buffer concentration can influence all these models, but at least this is the only data we have are accepted. Yes, the eukaryotic cell nucleosomes are packed into the chromatin structures, and the model is zigzag model. It's the most acceptable model.